to thank Dr. Pilar Muñoz Calero and also Angela Pico and Dr. Elisabetta.
For this communication, and I don't speak Espanol well enough. So I'll speak in Italian, slow, and perhaps in English for a part of communication. I work in experimental medicine department in University La Sapienza of Rome, and perhaps 4 or 5 years ago abbiamo iniziato
we decided to go into the field of MCS, especially thanks to the genome diagnostics, cooperating with the Department of Molecular Biology of the university with Professor Zimaco and the lab of Bilancio Radicalito with Dr. Chiara De Luca, who couldn't be with us today. And she has helped me to talk about neurology, endocrinology in relation to this field, and different biochemical mechanisms. And I'm going to present our findings from the point of view of the diagnosis in the patient. And there are recent new findings that may be of great interest. And we've checked out the involvement of the cytochromes, specifically in MCS patients, much more than in normal patients, in inverted commas. And this is very, very significant to me, because as you may know, multiple patients with MCS usually are sent to the psychiatrist. And this has severe implications because they don't get the right pharmacological treatment. And they get psychopharmacology that renders their symptoms more severe. In 2009, 2010, we saw the metabolization of these drugs, psycho drugs that lead into side effects. And these cytochromes do not help the patient to metabolize the drugs they're given. These patients, therefore, they cannot bear these, they cannot tolerate these psychodrugs. We have these catalase, and we have these glutathione as transferase levels. These two elements come out that are involved in this lack of tolerance. The SOD at the beginning in this study didn't show quantitative significant differences in terms of enzymatic activity. We later on saw genome-based differentiations. We had seen significant differences in terms of the glutathione. We had compared the enzymatic activities in patients, selected patients that were selected according to the Cullen questionnaire, and patients that had MCS-like symptoms. There were differences, basic differences in terms of the catalase and the glutathione. This is a chart that illustrates the existing differences between the MCS patient and the normal patient in terms of the null genotype with the methyltransferase 1 and significant differences you may see that indicate that in patients with MCS there is a much more common deletion than in the normal patients. Deletion of M1 means a loss, important loss of the enzymatic activity. In a normal individual, it wouldn't come up. So this is significant. And as you may see, there is a remarkable frequency in the MCS patient of a polymorphism of the SOD versus normal patients. At the beginning, we saw that the level of enzymatic activities didn't bring up many differences. When we dose the enzymatic activities, we have to look at the molecule level without knowing the function of the molecule that in many cases has to do with epigenetic actions. Most of the etiology in the environmental illnesses have to do with epigenetic factors, with transcription interferences or post-transcription interferences that prevent the construction of a good functioning enzyme that, although the gene structure is normal. And then in Italy, we wanted to see what mechanism is involved. In terms of the catalase polymorphism, we think that in terms of the catalase levels in the MCS patient, perhaps this enzyme works and functions and should be functioning. This is why it is involved in the cell mechanisms. We did research into the structure of the cell membrane to study the membrane in the inflammatory processes that are evident in MCS. And we saw, thanks to investigations on the red blood cell membranes, that these patients presented a high level of omega-6 versus omega-3, much more evident than what we see in normal patients. We know that we should have omega-6. In MCS patients, this is a high evidence. There must be a food restriction. In spite of having had a food restriction. and in the processing of the fatty acid analysis, we found baseline levels of the linolenic acid and the eicosanoic coming from the linoleic acid from the enzyme, the delta-6 desaturase, that is blocked in the oxidative stress. So there was much more pro-inflammatory eicosanoids. And there's a slight difference in terms of the saturated fatty acids, although without pathological meaning, because in a stressed body, the liver obviously will produce much more cholesterol, because cholesterol is at the basis of the production of steroids. And a stressed body requires much more of these, the steroid hormones. And a lot of interest is now on this field. There's a lab of biophotons started this year by Dr. Leopoldo Silvestrone, biophotons. And in this lab, something was discovered. Probably the biochemical mechanism we're talking about possibly could be the tip of the iceberg of the problem, because the intercellular communication doesn't only come through the biomechanical processes, the endocrine system, or the paracrine system, or the autocrine system, modalities that have to do with chemistry, but also may come from transfer of electromagnetic energy. This concept is a new one. It's in the field of the physics, belonging into the physical law, and in Stanford, Richard Feynman is bringing the interest of the researchers society. The Society of Researchers are interested in the quantic science, with physicists, doctors, physicians, chemists, biophysicists, and now they are brought into the field of the quantum entanglement. Entanglement belongs into the quantic energy field. It is a word coming from that field, entanglement. It is a mathematically based concept, mathematically demonstrating that reality isn't local only. Reality depends on time and space. So the cells communicate thanks to electromagnetic radiation. And Dr. Joachim Fisch in Germany and Fritz Albert Popp are behind this piece of research, and also someone in Russia as well. I'm sorry, I haven't put up his name here, who started doing research into this field back in 1930s. So Fritz Albert Popp tells us cells communicate through photon radiation. And the cause of this photon radiation has to do with the DNA. It is like a transmission network. Only 10% of DNA is known in our genes. Some time ago, people talked about DNA as spazzatura. The remaining 90% of the DNA has an unknown function. But it is necessary for the DNA to produce electromagnetic radiation in the cells. Generally between 200 and 800 nanometers with a low frequency. It's in a very low frequency order, 2, 7, 8 hertz. Generally speaking, we know that more closer we are to ultraviolet levels, it will be much more pathological. The important matter here is that indeed this communication network mechanism has been demonstrated by Fritz Popp. It has been reproduced using a group of homogeneous cells, especially lymphocytes, organized in two Petri dishes, separated one from the other, thanks to a membrane, a quartz-based membrane, a kind of a waterproof membrane to allow for different experiments. After following the cellular activity thanks to a mass spectrometer, in order to see on a monitor set the expression of proteins on the membrane of the lymphocytes, the two cell groups had the same behavior. Only one of the cell structures had a variation that had a different chemical stimulation as a function of time, producing a variation in the expression of the proteins on the membrane surface, a variation that didn't take place in the other cell group unless we maintained the dividing wall. The whole of the dividing wall, generally speaking, in some seconds, after some seconds without the stimulation, the other cell group would produce the same membrane protein. The only possibility of communicating across the membrane would be of electromagnetic nature. In order to explain this in the practice, there are different schools of thought. Some tell us about quantum energy. The brain, the neurons can communicate not only through neurotransmitters, but also thanks to photons, the physical state, based upon the mathematician Roger Penrose and the anesthesiologist Stuart Hameroff. There would be quantum neurorelationships. This is Penrose. Well, he's younger in this picture. And this interrelationship could have to do with the microtubules. that are the cytoskeleton of the cell. And many of our physicians think that they assemble and disassemble at very high speed, at a quasi non-local speed. And when they transport proteins, they don't require microtubuli. They can be kind of mouths. The proteins will be transported across the membrane, over the membrane, but not across it. And when there are microtubuli involved, this has to do with the electromagnetic transfer, the photons into the nucleus, into the DNA, and back outside the cell. And this fact may be demonstrated in this microphotograph of Giuseppe, and it has been photographed in order to see the wealth of microtubules present. Emilio del Giudice, Dr. Monroe knows him, knows Emilio. He demonstrated that water in cytoplasm, water makes up 90% of the membrane, and water is present in 99% of the molecular specimens. Quantitatively, 1% belongs to the rest of the molecular biology, but 99% is made up of water. But water isn't a chaotic liquid, isn't a chaotic fluid. The hydrogen may change the angles versus oxygen. And as a matter of fact, this is producing different coherence domains. And these domains of coherence are architecturally organized. And they represent water inside cytoplasm. When electromagnetic radiation moves across cytoplasm, as a function of intensity and frequency, it will go through the domain of coherence in a uniform manner. Or perhaps it wouldn't be powerful enough to move across. And there would be a resonance problem. 200, 800 nanometers, a low frequency level, 7 hertz. This electromagnetic radiation becomes a filament, microtubule-wise, producing electromagnetic radiation, building electromagnetic microtubuli in order to produce microtubulin that is part of the cytoskeleton. And through the microtubuli, the electromagnetic radiation would go through to reach the DNA. And once the function is accomplished, then the microtubuli will disappear and will be reassembled in a different way later on. We've seen a very little number of patients in a study that we want to do. Next year, we hope, in our center. We've seen that the MCS patients in general have electromagnetic emission of photons by their lymphocytes lower than the electromagnetic radiation present in normal lymphocytes. And, well, it's an observation. So we've seen that the stimuli, the sound stimuli that induce changes in the cellular electromagnetic radiation changes the status. In one patient was subject to a special type of singing that concentrates the vibrations at the vomer in the nose. In terms of a parallel pathway, parallel to the olfactory nerve. This would be vomer nasal specialized cells, an afferent pathway that belongs to the limbic system that in the mammals leads to reproductive function. These neuron cells have to do with the pheromones. And in human beings, the reproduction function has changed over 1,000 years, and these vomer nasal cells are atrophied in a large percentage of human beings. But occasionally, these neurons might be turned on. For example, in our own caseload, many patients with MCS had a blow on the nose that required surgical intervention. And perhaps this turned on the vomer nasal cells. And with this technique, we could see that there was an improvement in the emission of photons by the lymphocytes in MCS patients, thanks to this special type of harmonious singing. MCS would be the fruit of electromagnetic interferences that are common in industrialized countries. When I discussed this matter with my Chinese colleagues that study traditional Chinese medicine, I could get a report that in China, MCS has to do much more in those areas where there is electromagnetic preponderance than chemical preponderance influence. And electromagnetic interferences would be present interfering with the domains of coherence in the water of the cytoplasm and will be producing specific interferences in the biochemical cellular communication. We hope that this finding could bear fruit in the coming months, because we want to pursue this finding in future, this line of investigation. And we hope that you could understand my words.